So welcome to video one in our course G06 on RNA-seq and transcriptomics. This is going to cover RNA-seq what and why, the kind of central ideas behind it from different perspectives. So in terms of the procedures behind RNA-seq, you'll be familiar with this idea that we have various samples of interest. They may lack a condition that others have. We want to compare these condition one with the tumor to condition two which is normal. We isolate our RNA, which is quite fiddly, and we know that our RNA can degrade during that process. We do various library preparation steps, including poly A tail extraction, and then we are able to convert these RNAs to cDNA, add linkers and other pseudo markers relevant for our sequencing approach. We can then sequence them, take our reads, and do a mixture of de novo assembly and read mapping to determine the isoform types and levels that are present. And what we have to consider then is our experimental design, our samples, how we're going to collect our samples, how do they reflect the real world as much as possible? How do we isolate the mRNA? How do we convert it to cDNA? How do you prepare the libraries relevant for a sequencing approach? How is the sequencing conducted? How many cycles or read lengths is sufficient? We'll go through QC in the practical and read mapping, de novo assembly, counting our reads, quantification of our different isoforms. That will lead us then to be able to compare different groups if that's our purpose. So that's kind of our detailed steps. Our rough outline is something we can think about as well. We have RNA that's of interest. We prepare our libraries. We sequence it. So remember that we're losing information through this process. We then try to estimate the abundances of the different isoforms that are present and compare different groups. So that's kind of more short version that we're familiar with. And we're going to focus on this part where we're trying to take the sequencing data in the context of the experimental approaches that have occurred before to appropriately apply to relevant models that will tell us more about the isoforms that are found and also their levels. So I guess a key difference for RNA-seq that we should bear in mind is that we're mapping reads or used to mapping them to a genome with typically exons and introns. So exons should have lots of reads from mRNA-seq data, depending on RNA protocol, which may be specific for non-coding RNAs as well. There are different protocols. We then have to consider if looking for mapping to a genome or a reference transcriptome. So that will give us a slightly different picture, but also will identify different reads that map across different exon boundaries. So these splice junctions means that we have to consider what kind of reference we're using and how our mapper works. So if you think about those things in the context of what's our goal for looking at this RNA-seq data, and those goals can be quite diverse in this area. Most of the time we're looking at mRNA sequences. So in that context, we're trying to think about we have some read. I want to think out where did this read come from, and can we identify the exact position from where it originated. And then we have to consider the experimental context that different sequencing experiments will be on different days with different amounts of inputs, cDNA, different library preparations, different artifacts, which means we have to think about, given the diverse sources of different reads with different levels, how do we normalize them appropriately in a manner that means that they're meaningful when we compare them against each other, and solve this problem above. Get the number of reads associated with each CDS and locate them precisely within that CDS. That's our challenge. So RNA-seq is different to microarray experiments, and RNA-seq is essentially better. It's more sensitive and specific to detecting RNAs in general. And there's been copious studies from 2015 onwards showing that RNA-seq is more accurate for identifying differentially expressed genes and is very limited technical variation in general. So that definitely outperforms microarrays, which are now more historical. And that's really been demonstrated in many different studies showing that they're more effective at detecting low abundance transcripts or novel isoforms as well in terms of assembling different alleles that might be present. And also show broader dynamic range than microarrays, and we don't have to know the transcripts that might be expressed in advance. So for all these regions, RNA-seq is much, much better. And it's also worth noting that RNA-seq approaches can improve gradually over time as well. It's not a single static entity. We can also think about different ways of analyzing RNA-seq data. And there's two central approaches in order to the models that are applied, one of which are count-based models, which are kind of shown in red here, and some of which are linear model-based. So the likes of lemma would be down this angle. So there are similar aspects at the start that we need to scale our data appropriately and transform it. They all share gene filtering steps where we might have many genes with zero or no data available. There may be some transformation steps involved. And the central difference will be that count-based models consider their data as being model-based in a negative binomial distribution. So they're looking at the individual counts that are observed and trying to estimate the expression levels across different samples in different contexts, estimate the variation we have for each particular isoform that is present, and try to link these all together to compare, say, different groups or look at the quantitative distribution of input phenotype data to which we want to link this gene expression data. Then we have lemma, where we're looking at the kind of some one linear model with some sort of parameters or weights that inform our model, and they may be present and absent for different groups that are of interest that we can then compare. Obviously, if we have 20 or 30,000 different genes and even more isoforms, we have to consider multiple test correction. And then also, if we're looking at say the dispersion and shrinking that dispersion, what kind of models are we applying here, and are they effective? So if we go back through the history of RNA-seq, one of the first initial tools that was used most widely was called Bowtie. So this was published in a paper, Ultrafast and Memory Efficient Alignment of Short DNA Sequences to the Human Genome. And what was interesting about that is that it aligns the reads without gaps. So this was an initial paper to kind of set the scene for how we can achieve this challenging issue of mapping RNA reads to a reference genome. And that used near exact matches between the 5' ends of known as the C region of reads and the reference genome. So for efficiency, it just looked at the first 28 bases to assign a read to a location, and that kind of approach was used more widely in many different approaches. So the issue was that exact matching can be memory intensive and take time. Improvements were made with a version of it called Bowtie 2, and that allowed gap alignments and allowed gap opening and penalties and so on. So that's two stages where it was taking that seed, looking for a ungapped alignment, and then using kind of an extension, a bit like the last extension steps, to kind of expand that matching area. And then those other approaches, Bowtie 2 was superseded by HiSat2, the hierarchical indexing first place alignments of transcripts. But now that's a bit old, and there's better approaches using Minimap 2, which is a much better aligner now. There's also other methods that are now historical or were used very widely at the time, DSeq and DSeq2, HR as well. But we know now that these methods have high false positive rates, so they're not advised. So that's our short background into the wider scope of RNA-seq, and we'll talk about more specific things in our next